Now, having understood reversible cell injury and how the two deciding factors make it go into irreversible cell injury, let's understand the mechanism of what is happening here. In irreversible cell injury, I just told you that there are two factors. One, severe membrane damage, and two, severe mitochondrial damage. Now, when membrane is damaged and mitochondria is damaged, we say it is irreversible. Let's understand that. When the membrane is severely damaged, there will be something from outside that will enter the cell. Ideally, everything can enter because that membrane is gone, but you need to understand what is the important thing that is entering is calcium. Now, just a concept for you to hold is that all the calcium that you have in your body, all of it or majority of it is extracellular. The body tends to keep it outside the cells. And why is that so? Because if the calcium would be kept inside the cells, it will activate all the enzymes, and all your enzymes after getting activation will destroy your cells. We don't want that. We don't want the cells to get destroyed by the own enzymes. So we keep the calcium outside, we keep the enzymes inside so that this stays separate. But now what is happening? The membrane is getting damaged and there is going to be calcium influx. What will this calcium do? This calcium will then go and activate the enzymes. Which enzymes? It will activate the phospholipase, it will activate the protease, and it will activate the nuclease. The phospholipase, protease, and nuclease. So the three building blocks of the cells, the phospholipids, the proteins, and the nucleus. All the enzymes targeting them are going to get activated because calcium has come inside. Correct? Also, this calcium will then go and deposit inside the mitochondria. And this will result in the formation of something known as amorphous densities. It will result in the formation of amorphous densities over here. Now, there are questions about amorphous densities that you need to know. Number one, what is the composition? So remember, the composition is calcium. You know that. Number two, you will be asked, which organelle is an amorphous density formed in? And I told you our favorite organelle for this chapter is mitochondria. Answer to most of the organelles will be mitochondria. And question number three that you will be asked, which injury does it appear in? So remember, we've studied it under irreversible cell injury. So amorphous densities is a sign of irreversible cell injury. I want you to recall myelin figures were appearing in reversible cell injury, but their number increases in irreversible cell injury, but they first start appearing in reversible. Amorphous densities appear in irreversible cell injury. They are not present in reversible cell injury. So now what happened? Because of this formation of this amorphous densities, I can say that there is severe mitochondrial damage. Remember the two deciding factors of irreversible cell injury. First, severe membrane damage, all the calcium enters. This calcium then goes and destroys the mitochondria. So severe mitochondrial damage, both our factors are done. Before we proceed, I want you to focus on the term that calcium activated these enzymes like phospholipase, protease, and nuclease. What additional do you need to know about this nuclease? The nuclease will go and break down the nucleus, right? That is obvious. The nuclease will go and break down the nucleus. But now nucleus has all your genetic material. It has DNA. It is a complex structure. One enzyme cannot just go and break it in one go. So this breakdown will have certain steps. What you can also call as nuclear changes. And what are those? So for that better understanding, let's consider this to be a normal cell, right? And within the normal cell, I have a normal nucleus. So this is a normal cell. Now what is happening is, let's say the first change. Again, I'll draw a cell. The first change is the same cell I have, but now focus what has happened. Do you see how the size of the nucleus has become smaller? Number one, the nuclear size decreases. And do you also notice how I've drawn a darker shade? It becomes dark blue, that is, it becomes hyperchromatic. It becomes hyperchromatic. This change, the first change, this is known as pycnosis. So when the nucleus starts to break down, first it undergoes pycnosis. That is, the nucleus is going to become smaller and more dark in color. That is pycnosis. After pycnosis, we now enter into the second stage. So let's see what is happening in the second stage. Again, we have this cell. But now please note how the nucleus is a little different here. Do you see that now the nucleus has been broken down into pieces? This, students, is the second step referred to as karyorhexis. Please focus on the term. Karyo means nucleus and rhexis means fragmentation, right? So this is nuclear fragmentation. This is nuclear fragmentation. This is the second step. The third step that we go towards now, students, is going to be the final step where the nucleus is gone. It has completely vanished. That is referred to as karyolysis. So you can again focus. Karyo means nucleus. Lysis means dissolution. The nucleus has dissolved. That is referred to as nuclear dissolution. So now if you are asked that the nuclear breakdown or the nuclear changes that take place in irreversible cell injury, please remember in irreversible cell injury, you need to remember this particular order. That is, we have pycnosis, then there is karyorhexis, and then there is karyolysis. On this order particularly, you have been given questions many a time. Pycnosis, karyorhexis and karyolysis. So this was about irreversible cell injury. Remember, severe membrane damage, severe mitochondrial damage, everything is related to calcium. Finally, the phospholipids, the proteins, and the nucleus will break. The nucleus will break in 3 steps. That is number 1, pycnosis, number 2, karyorhexis, and number 3, karyolysis. If you go back to your main flowchart, with this we finished irreversible cell injury. So we are now beginning with what we know as cell death.